Hello and welcome. In this video we will discuss exercise 13, which includes lung volumes and capacities. So specifically what we'll talk about are the basics of the form and function of lungs. We'll talk about the law that describes the relationship between volume and pressure of a gas. We'll talk about breathing, and then how we study breathing, and then how to read data for this week's lab exercise. So the lungs, they are very important to us because they're what bring in the oxygen to our blood, and they're also what helps us to get rid of carbon dioxide from our blood. You want to think of these as very elastic tissue, but they're very thin, so thin that in the pressure of our atmosphere they would want to collapse. And so the reason they don't collapse is they're inside of what's called our thoracic cavity, which is what houses our interpleural space. And so it's actually inside that interpleural space where our lungs sit, and that's a negative pressure environment. So in that negative pressure environment, they do not collapse. But we can get them to collapse if, say, we punctured a hole in the thoracic cavity, and that would cause the pressure inside to equal the pressure outside, and then the lungs would collapse in a condition that's called pneumothorax. A little bit more about the lungs. There's two cell types worth noting inside the lungs, type 1 and type 2. Type 1, those are the by far most numerous, so those are what line all the alveoli. These are think of them as all these very thin sacs that are adjacent to capillaries. So this is where gas exchange occurs in our lungs. So there's a bunch of those that sort of form those alveoli, the lining of the inside of the alveoli, and then there's type 2, and what these do is secrete something called a surfactant that prevents our alveoli from collapsing. It's a soapy type liquid, so helps to make bubbles or keep what is essentially a bubble, our alveoli, from popping or collapsing. Surfactant isn't produced until late in development, and so sometimes premature babies are born without that surfactant, and so they need help keeping their alveoli from or their lungs from being collapsed so that they can breathe when they're born too early. Okay, so let's talk a little bit more about breathing. It takes advantage of this law that describes the relationship between volume and pressure of a container. So if you decrease the volume, then you increase pressure, and if you increase volume, then you decrease pressure, or this equation that we know as V1 P1 equals V2 P2, or volume 1 and pressure 1. Okay, so what does that look like in the lungs? Well, when we inhale, inspiration, this is an active process, and so this is largely regulated by what's called our diaphragm. You can see this blue line here. It's a muscle. It forms the bottom of our thoracic cavity, and so to inhale requires contracting that muscle, which causes it to drop, and when the diaphragm goes down, then the volume inside the thoracic cavity increases, and this causes the space around the lungs to increase as well, and so the pressure there starts to drop, right? If we increase volume, we decrease pressure, and then that causes the lungs to expand, and so their volume increases, and then the pressure inside the alveoli goes down, and so when the pressure inside is lower than the pressure outside, then the outside pressure comes in, or it goes from a higher to a lower pressure, and so that's how we fill our alveoli with fresh air every time we take a breath. And then the other side of that, it's not an active process, what we call expiration. This is typically passive. It doesn't require energy. In fact, if we just stop flexing our diaphragm, then it kind of goes back up as we relax, and the pressure of the flexibility of our rib cage and the intercostal muscles, the elasticity of those just kind of causes our thoracic cavity to get smaller. And then we exhale, and so when we exhale, we decrease the thoracic cavity volume. And so remember, when we decrease volume, we increase pressure, and if the pressure is higher, then it will go to the lower pressure and it will leave our lungs. So you could just take my word for it, or you could watch this brief video right here. So what's going on in this video is what we see is this chamber, and at the bottom is a diaphragm, or what simulates a diaphragm, and the balloons simulate the lungs. And notice as the diaphragm drops, the space inside increases, or the volume inside increases, and so the pressure decreases, and that causes those balloons / alveoli to fill up. And then as the diaphragm comes back up, the volume gets smaller, the pressure increases, and the air leaves the balloons / alveoli. So here's the good stuff that you really want to know for this week's lab, and this is how to understand lung volumes, or the study of what we call spirometry. So what we're looking at here is several things related to lung volumes and capacities. So let me just point out what we need to know about what's going on here. So if we follow this black line, this sort of shows our breathing volume in and out. As it descends, it's going out. So this is what's called tidal breathing. This is your typical breathing capacity. It's about 1/2 liter for adults. That's how much air you're moving in and out during, let's say, when you're watching TV or watching this video. And next we have the inspiratory reserve capacity. And so that's the amount of air that you can inhale or inspire in addition to your normal tidal inspiration. So that's like as much as you can breathe in. And then following that, we have the expiratory reserve capacity, or reserve volume, here on this diagram. And so then that's the amount that you can expire or exhale following a regular tidal exhale. So that would be like getting all the air out of your lungs. And the volume for that is around 1,200 mils, whereas the volume for inspiratory reserve is around 3,000. And then next we have something that's called residual volume. So residual volume, that's the amount of air that's left in your lungs even when you've exhaled as much as you can. So notice you would have to completely collapse your lungs to get all the air out. So you can't breathe it all out. So there's a little bit left in there. We call that residual volume. That's also around 1,200 mils. And basically that's the volume that keeps your lungs from collapsing. And then next we have something that's called vital capacity. So that includes your inspiratory reserve, your tidal volume, your inspiratory reserve, your tidal volume, and your expiratory reserve. So that's really important, because that's how much air you can move in a given breath, maximum in and maximum out. And so that's important for determining pulmonary function and things like restrictive lung diseases. And then something else that we can determine from this, if we track the time, is what's called the forced expiratory volume, or the FEV. And that's the total amount that you can exhale after a maximal inhale in 1 second. So how much can you get out, and how fast can you do it? And that's going to be a good indicator of obstructive lung diseases. So let's take a look at those in a little bit more detail and see how they relate to our lab activities. So in lab this week, we'll be measuring tidal volume, we'll be measuring vital capacity, and we'll be measuring forced expiratory volume. So we want to compare those to normal and then see what that indicates about the individual. And if the data shows that it's lower than normal, then this might indicate restrictive lung disease. meaning there's a loss of elasticity and the lungs are restricted. So this might be something like pulmonary fibrosis, where there's scarring of lungs, the lung tissue, so they can't stretch as much when breathing in, respiratory distress syndrome, or something like emphysema. So the other thing that we'll be measuring is forced expiratory volume, which is, again, the amount of air that can be expired during a single second. So normal people should be able to get about 80% of that air out in a second. Borderline would be between 60 and 80, and disease would be 60. And the disease in this case we're referring to is obstructive lung disease, and that would indicate something like the air is getting through, or the air is being blocked on its way through, or the tube it's getting through is being narrowed. So maybe it's inflamed, or maybe there's a lot of mucus, or maybe you've got a peanut stuck in your throat. But that's going to decrease the rate at which air can move through. So asthma and emphysema, again, are examples of obstructive lung disease. So data this week, it'll look a little bit like this. Remember to use these icons to highlight a single wave, and then you can zoom in on it so that you can measure the amounts and time in a precise way. And as you highlight, your data will appear down here. Data which was gathered by an airflow transducer that was hooked up to a BioPack machine, measuring the amount of air moved by the person that was breathing in and out of the tube. Thanks.